Why is it so important to understand the structure of HIV and its interactions with antiretroviral drugs? Researchers study this in order to enhance drug effectiveness that target HIV's proteins that are crucial for its survival. HAART, or highly active antiretroviral therapy, is the culmination of the following drugs. Generally, these drugs mimic the enzyme's normal substrates and inhibit proper functioning. HIV protease inhibitors target HIV-1 protease, which is responsible for cutting the virus's long polypeptide chain into functioning protein-sized pieces. This enables the virus to mature and infect new cells. HIV-1 protease contains two flaps that wrap around its substrate at the active site, where the polypeptide is cleaved by a water molecule. These inhibitors, such as the drug saquinavir, mimic this protein chain and block the active site. Since these inhibitors cannot be cleaved, they prevent the normal functioning of this protein. Reverse transcriptase inhibitors target reverse transcriptase that builds the DNA copy of the viral RNA genome used to create new viruses. There are two types of drugs that target this protein. One type is called NARTI's nucleoside analog reverse transcriptase inhibitors, which is a modified nucleotide without a connector, such as the drug AZT, which halts DNA synthesis. The other type of drug, called NNRTI's non-nucleoside reverse transcriptase inhibitors, changes the shape of the protein's active site and prevents DNA from binding. Another target protein is integrase, which incorporates the DNA copy of the viral genome into the host DNA. Antiretroviral drugs such as raltegravir bind to the enzyme's active site and displaces one end of the DNA strands, thus inhibiting enzyme action. The last set of drugs target CCR5, a chemokine receptor that acts as a doorway for HIV to inject its viral components into a host cell. First, GP120 on the viral cell attaches to the CD4+ receptor on T cells and undergoes a conformational change. At this point, the nucleocapsid can enter into the interior of the cell. Drugs such as maraviroc bind to the receptor and prevent viral proteins from attaching and entering a cell. Through studying the structure and interaction of HIV with the current antiretroviral drugs, we are one step closer towards eradicating this lethal disease.